climbs a blade of grass. It reaches the top, clamps its jaws around the stem, and does not let go. It did not choose to climb. There is no food up there. The colony does not need it at this height. Nothing about the ant's normal behavior explains this final act, except that something inside the ant made the decision for it. The ant is still alive. Its legs still twitch. Its body still responds to light and temperature. But the behavior, the climb, the bite, the location, the timing, belongs to something else. Most people, when they think about the worst ways an animal can die, imagine being caught, chased down, seized, torn apart. The threat arrives from the outside, and the encounter ends in death. That is a story with two participants. One wins, one loses. The relationship is violent, but it is simple. Parasites do not follow that logic. A parasite does not always kill its host, not immediately. It enters the body, and instead of destroying it, it uses it. It feeds from the inside. It grows. And in some of the most studied cases in biology, it changes what the host does. Not randomly, not as a side effect of sickness. Specifically, precisely, in ways that benefit the parasite and nothing else. The body keeps moving. The animal keeps breathing. But the instructions have changed. It happens across dozens of species, on every continent, right now. And the question it raises is one most people never sit with long enough. What is a body, once the decisions it makes are no longer its own? The fungus is called Ophiocordyceps unilateralis. It infects carpenter ants in tropical forests across the world. The infection begins when a spore lands on the ant's exoskeleton. The fungus penetrates the cuticle and enters the body cavity. Over the following days, it grows through the ant's tissues, consuming non-essential cells first, keeping the animal alive and functional for as long as possible. Then the behavior changes. An infected ant abandons the canopy trails where its colony forages. It descends. It moves to the underside of a leaf, roughly 25 centimeters above the forest floor, a height where temperature and humidity are optimal for fungal reproduction. It bites down on the leaf vein with a force researchers have described as a death grip. The mandibles lock. The ant does not release. Then the fungus kills it. A fruiting body erupts from the back of the ant's head. A stalk, sometimes longer than the ant itself, grows upward and releases spores into the air below, directly onto the trails where other ants walk. The precision is what disturbs researchers. Not just that the ant climbs, not just that it bites. The location, the height, the orientation of the body, the timing of death relative to spore maturity. A 2009 study in a Thai rainforest found that infected ants consistently bit onto leaves at a specific compass orientation, at a precise height range during late morning, conditions that maximized fungal reproduction. The behavior did not vary. The pattern did not drift. The fungus had effectively programmed the host's final hours. And here is what makes this harder to dismiss as simple sickness. Researchers have found that the fungus does not appear to directly invade the ant's brain. Tissue studies show fungal cells surrounding the muscle fibers, particularly in the mandibles, but not penetrating the central nervous system in significant numbers. The ant's brain remains physically intact. The control appears to come not from hijacking the brain, but from manipulating the body around it. The ant's muscles are being operated. The brain may still be sending signals, but the body no longer obeys them. Ophiocordyceps is dramatic. It is not unique. It is one example in a pattern so widespread that biologists have a term for it: parasite-induced behavioral manipulation. The strategies differ enormously. The outcome does not. The jewel wasp, Ampulex compressa, uses a cockroach, not as food for itself, as a nursery for its offspring. The wasp stings the cockroach twice. The first sting targets the thoracic ganglion, temporarily paralyzing the front legs so the cockroach cannot fight back. The second sting is surgical. The wasp inserts its stinger directly into the cockroach's brain, specifically into regions associated with the escape reflex. Researchers have shown that the venom contains a cocktail, including GABA, and a GABA receptor agonist that does not paralyze the cockroach, but suppresses its motivation to move. The cockroach can still walk. It can still right itself if flipped over. Its motor systems are intact, but it will not run. It will not resist. The wasp grabs the cockroach by its antenna and leads it, walks it like a leashed animal, into a burrow. There, the wasp lays an egg on the cockroach's leg. Then it seals the burrow shut. The cockroach stays alive, docile, and metabolically active while the wasp larva hatches, chews into the cockroach's body, and feeds on its organs in a specific order, consuming non-vital tissue first, keeping its host alive and fresh for as long as possible. The cockroach is not dead. It is not unconscious. It has been chemically prevented from caring. Hairworms push the manipulation in a different direction entirely. The parasite Spinochordodes tellini infects crickets and grasshoppers. It grows inside the insect's body cavity, feeding on nutrients in the hemolymph until it reaches adulthood, at which point it can be several times longer than the host. But hairworms are aquatic. They need water to reproduce. Crickets avoid water. Under normal conditions, a cricket that encounters a pond or stream moves away. Water is death for a terrestrial insect that cannot swim. Infected crickets walk toward water. They jump in. Studies published in Proceedings of the Royal Society have shown that the worm produces proteins that mimic the host's own central nervous system molecules, effectively altering the insect's neurochemistry to reverse its water avoidance behavior. The cricket does not stumble into the water by accident. It seeks it out. When it drowns, the hairworm emerges from the body and swims free. The host's deepest survival instinct was not damaged. It was overwritten. But reversing behavior is not the only option available to a parasite. Some do not just change what the host does. They change what the host is. The flatworm Leucochloridium paradoxum infects amber snails. Its larvae migrate into the snail's tentacles, where they form swollen, pulsating sacs striped in green and brown. The infected tentacles no longer look like tentacles. They look like caterpillars. They pulse rhythmically, a motion visible from a distance, mimicking the movement of insect larvae. Snails are normally cryptic. They avoid open, well-lit areas where they would be visible to predators. Infected snails do the opposite. They move into exposed positions in brighter light during daylight hours, behavior that makes their throbbing, caterpillar-mimicking tentacles visible to birds. A bird eats the tentacle. The parasite reaches the bird's gut, completes its life cycle, and its eggs pass out in bird droppings, where another snail will eventually encounter them. The snail is not killed outright. It can regenerate the tentacle. The brood sac can reform. The cycle can repeat. The snail becomes a reusable lure, its own body reshaped into bait, positioned by altered behavior in exactly the place where a predator is most likely to strike. The parasite did not just change what the snail does. It changed what the snail looks like. The lancet liver fluke, Dicrocoelium dendriticum, requires 3 hosts to complete its life cycle. It begins in a land snail. The snail excretes slime balls containing fluke larvae. Ants eat the slime balls. The larvae migrate through the ant's body, and one, typically just one, settles near the subesophageal ganglion, a nerve cluster that influences the mandibles. At dusk, when temperatures drop, the infected ant leaves the colony. It climbs to the top of a blade of grass. It clamps down and waits. If a grazing animal, a cow, a sheep, a deer, eats the grass, the ant enters the digestive system, and the fluke reaches its final host, the mammalian liver. If no grazer comes, the ant releases its grip in the morning when temperatures rise. It returns to the colony. It behaves normally during the day. It forages. It follows pheromone trails. It functions as a colony member. Then, at dusk, it climbs again. The fluke does not destroy the ant. It turns it into a repeating delivery mechanism. Night after night, the same behavior. Climb, clamp, wait, release, return, climb again, until the ant is finally eaten, or until the fluke inside it dies. Each of these cases uses a different mechanism. Chemical manipulation of muscle tissue, targeted venom delivered to a specific brain region, molecular mimicry of host neurotransmitters, direct interference with a nerve ganglion. The strategies are distinct. The result is the same. The host's body serves the parasite's life cycle: movement, feeding, fear, reproduction, timing of death, whichever behavior the parasite needs redirected. Evolution has found a pathway, and the pattern does not stop at insects. Toxoplasma gondii is a single-celled parasite that can infect virtually any warm-blooded animal, but it can only reproduce sexually inside the gut of a cat. Its life cycle depends on getting from an intermediate host, usually a rodent, back into a feline. Infected rats show reduced fear of cat urine. Some studies have found that infected rats are not merely less afraid of the scent, they appear drawn to it. Research has shown that Toxoplasma infection specifically alters the rat's response to cat odor without broadly suppressing fear of other predators. The parasite did not make the rat fearless. It made the rat less afraid of the one predator the parasite needs. The mechanism appears to involve cyst formation in the brain, particularly in the amygdala, a region involved in fear processing, and shifts in dopamine and testosterone levels. The exact pathway is still debated. Some researchers caution that behavioral changes observed in laboratory-raised rodents may not fully mirror wild conditions. The science is active. The pattern is consistent enough to have generated hundreds of papers across more than 2 decades. Toxoplasma also infects humans. An estimated 1/4 to 1/3 of the global population carries it, though prevalence varies enormously by region. Whether it meaningfully alters human behavior is one of the most contested questions in parasitology. Some studies have linked chronic infection to subtle changes in reaction time, risk-taking, and personality profiles. Others have found no significant effect. The sample sizes vary. The confounds are difficult to control. The claims run from cautious to sensational. What is not in dispute is the mechanism in rodents. A single-celled organism with no nervous system of its own alters the fear response of a mammalian brain, not through brute damage, but with enough precision to change which predator the host avoids. There is a parasitic barnacle called saculina that infects crabs. It enters the crab's body through a joint in the exoskeleton, then spreads root-like tendrils called an interna through the host's tissues. It consumes the crab's gonads. It takes over the reproductive system. A male crab infected with saculina stops developing male characteristics. Its body begins to resemble a female's. It will even perform the behaviors of a brooding female, fanning and caring for the parasite's egg sac as though it were the crab's own offspring. The crab does not reproduce. It cannot. Its reproductive organs have been consumed. But it tends the parasite's young with the same behaviors it would have used for its own. The body is still a crab. The life inside it belongs to something else. There is a pattern here that goes deeper than discussed. A predator treats the body as food. It ends the story. A parasite treats the body as infrastructure. It continues the story, but changes the author. Evolution does not require awareness. It does not require cruelty. It does not plan. It requires only that organisms which reproduce more effectively leave more descendants. If a fungus that redirects an ant to a precise leaf height releases more spores than one that does not, the redirecting fungus wins. If a wasp whose venom suppresses the escape reflex raises more offspring than one whose venom simply kills, the precise wasp wins. The manipulation is not designed. It is selected for. And the selection has been running for hundreds of millions of years. Hosts evolve countermeasures. Immune systems learn to detect invaders. Behavioral defenses emerge. Ants that carry infected nestmates away from the colony. Fish that rub parasites off against rocks. Primates that avoid contaminated food sources. The arms race is not one-sided. But the parasites that survive are the ones whose manipulation is precise enough to outpace the host's defenses. And precision, refined over geological time, produces results that look engineered, even when no engineer exists. Death is a straightforward transaction. The difficult part is everything before it. The interval when the body still functions, still walks, still appears to act on its own judgment, but the purpose of those actions has shifted to serve something else entirely. The body becomes a tool, a shelter, a vehicle, a nursery, a delivery system, a repeating trap. Parasitism is not a fringe strategy. By some estimates, parasitic species may outnumber free-living species on Earth. The majority of life may depend, at some stage, on using another living body. And in most of these cases, there is no evidence that the host knows. No documented moment of resistance where the ant tries to descend and the fungus forces it back up. No sign that the cockroach attempts to flee and fails. The manipulation works because it operates below the level of what the host can detect or oppose. The body does what it is told because the instructions arrive through channels the host has no architecture to question: its own muscles, its own neurochemistry, its own reflexes. Biologists do not call this mind control. The term is too imprecise, too saturated with human assumptions about will and intention. What they describe is behavioral modification through biochemical interference. The host's nervous system is not overridden the way a driver seizes a steering wheel. It is edited, quietly, from the inside, in ways the host's own immune system may never register as foreign. The question that remains is not whether parasites are cruel. Cruelty requires intent, and there is none here. The question is simpler and worse. How would you know if something changed the way you responded to fear? If something shifted, which risks felt tolerable and which felt deadly? If your body moved toward a place it would normally avoid, and the decision felt like yours, felt ordinary, felt unremarkable, felt like something you simply wanted to do? The lancet fluke does not make the ant suffer. It makes the ant climb, and the ant climbs as though climbing were its own idea. The body does not always belong to the thing living inside it. That is not a horror story. That is a biological fact, documented across phyla, across continents, across hundreds of millions of years of coevolution. And the most unsettling part is not that it happens. It is that the host never knows.